Okay, so thank you very much again for that introduction. It's a real pleasure for me to be able to talk to you today about the research that our lab is doing. And so I am going to talk about a couple of facets of the way that we're approaching our research. So that is using distributed drug discovery, and I'm going to explain what I mean by that in a moment. As well as drug repurposing strategies to really drive NTD drug discovery programs. And so I'm sure that you all are very familiar with this image. I know it's a few years old now, but there are a couple of points that I really wanted to use to demonstrate or to talk about around this. So obviously there are 20 neglected tropical diseases as listed by the World Health Organization. As Maya actually just mentioned, our real main focus of the focus of the lab is really on Chagas disease, human African trypanosomiasis, leishmaniasis, and schistosomiasis. Across this space, there are 2.3 billion people living at risk of infection, and an additional 1.1 billion who are infected with at least one of these NTDs. And so it's been, it's an area that really needs a lot more attention than it's currently getting. So firstly, approaches to repurposing. Why are we thinking about repurposing drugs? Firstly, it's well established to work well for NTDs. Eflornithine was is currently a treatment for HAT, but was originally a cancer therapeutic. Miltefosine for leishmania was originally also developed as an anti-cancer drug, and similar stories for chromomycin and amphotericin B. So these are all approved drugs that are currently being redirected or directly repurposed in their current state. And that really begs a lot of questions. So the first is, what is the potential for the toxicity? Are we settling for what we have instead of what we could have? And can we use drugs from the current pharmacopoeia as starting points for optimization? And as an extension of that question, how do we most efficiently select starting points to optimize and to work with as part of the lab? And so I'm going to touch on those briefly today as we go through this presentation. I'm sure all of you are familiar with the typical drug discovery pipeline, where we go from validation, compound screening, optimization, and then into clinical trials before nominating a drug. The advantage of repurposing, though, is that we actually kind of skip the first couple of stages, the validation and the compound screening. We identify a compound of interest that we can enter straight into the chemical optimization phase. And the question that we're asking ourselves here is, can we actually shorten these timelines? So by considering the information that we have out there in the literature, whether it be in terms of journal publications, patents, anything that was generated as part of the discovery process for these compounds, can we use that information to improve the potency of compounds, or rather to improve the selectivity of compounds, and so knock out human activity as part of our efforts, and thereby shortening these timelines. And so there's actually a really great review on this, and so I'm not going to spend too much time here. This was actually written by Dana Klug and Michael Polastri, and so Dana you'll be hearing from later today. And this is really kind of repurposing in a nutshell. So today I'm going to be talking about target class repurposing on the bottom right. And so really what we're doing is we're taking an approved drug. We do need to do further optimization, but the way that our lab works predominantly is via phenotypic screening. We don't necessarily understand the target of our compounds when we begin, and I'm happy to take questions about whether that's good or bad at the end. And so without further ado, I would really like to focus on the story at hand. So target class repurposing, this is a story about the optimization of lapatinib, which is a human EGFR inhibitor. You can see that there is our lab has been working on this for a number of years now, and we've been working with a number of different partners. And so I'm going to kind of try and loop together two stories here. So target class repurposing. From the very beginning, I posed the question, how do we identify compounds to optimize? So there is a lot of literature that describes trypanosome kinases. I'm just giving you numbers here. There are 176 kinases within Trypanosoma brucei, 190 within cruzi, and 199 within leash major. There were a couple of key observations. There are no protein or receptor tyrosine kinases. And non-specific tyrosine kinases actually block transferrin uptake in T. brucei and impact parasite growth. And it was actually thanks to a collaborator of ours, Dr. Kojo Mensah-Wilmot, who was at the University of Georgia and has actually just moved to Kennesaw State University, also in Georgia. They actually found that when they took lapatinib, so again, an approved drug used to treat breast cancer, they actually found that taking lapatinib, it was moderately potent at 1.5 micromolar, but it actually elicited a cure in terms of the in vivo efficacy study. We admittedly cured 1 out of 4 mice, so it requires further optimization. But this is just demonstrating this idea of actually focusing in on what can we target, what starting points actually make the most sense. And this was initially what our project looked like. We were doing the compound synthesis, the design work, we were testing at Northeastern, and then we were shipping compounds to the University of Georgia, and Kojo's lab was actually doing all of the in vitro screening for us. So we were very quickly able to develop the SAR, the structure activity relationships around this series, using a couple of strategies. So firstly, we focused in on each region of the molecule. We were able to design a virtual library, we were able to apply filters, and we went from a compound that NEU 369, you can see we've really only changed one region of this molecule. We've maintained potency against T. brucei, but what we've also done is improve our selectivity index. And so now we are actually at 50 micromolar against HepG2, which is a mammalian cell line. From there, we were able to make further modifications. We've increased our potency against T. brucei, maintained our selectivity, or actually improved our selectivity as well. And this process took us 44 compounds across 3 cycles to get to a compound which was vastly superior versus lapatinib. And this is kind of where our network really starts to grow. And so you can see here with our screening campaign, what we're now doing is we're actually now working with multiple biological collaborators. So what this means is, so again, the synthesis, all the design work is happening at Northeastern, but now we're shipping out compounds to 3 different labs. So to the University of Georgia, we're doing the T. brucei screening. We're also shipping to the New York University, where they're screening our compounds for T. cruzi activity. And we're also shipping to the Walter Reed Army Institute of Research, who are looking at leash major, PFAL, as well as measuring HepG2 toxicity for each of our compounds. And so we're now starting to generate an incredible data set of information that we can use in terms of optimization, not just for sleeping sickness. We're now also considering Chagas disease and leishmaniasis and malaria as part of our optimization. The compound that we had identified, yes, definitely more potent. Unfortunately for us, it wasn't very drug-like. When we consider what the target product profile for African sleeping sickness is, really we're looking for an oral drug that can be taken, that's very stable, and can really be kind of utilized within the community rather than requiring hospitalization of patients. And so our next part of this project really focused on improving the properties. And so I have color coded here for reference. So NEU 961, it was a really potent compound. We were quite happy in terms of its potency. But the really big thing for us was it was very lipophilic. It's basically a grease ball. It's also, because of that, doesn't have great solubility. And we really needed those two things to be improved for two reasons. Compounds that are not soluble, they're not going to be bioavailable. They will require some kind of formulation to actually achieve exposure, which is not what we're trying to do. We're really trying to make sure we can identify an oral therapeutic. And really, lipophilic compounds tend to be quite promiscuous. So in order to address this, we actually focused on what we're calling the head group region. This is our largest, most lipophilic portion of this compound, lipophilic meaning greasy, and it has the flattest SAR. And so we were actually able to generate a 246-member virtual library. From this, we could again apply properties filters. We were thinking about things like the rule of 5, the central nervous system multi-parameter optimization score. For those unfamiliar with the CNS and PO score, it's really thinking about identifying compounds that can cross the blood-brain barrier or have the potential, are predicted to have the potential to cross the blood-brain barrier, which is really important when we're thinking about stage 2 of HAT, which is obviously all within the CNS. So this led us to identifying a properties compliant virtual library. We then did a 3D shape and electrostatics comparison with our original biphenyl ether head group in red. And we identified and chose 14 analogs that we were going to synthesize. Ooh, there we go. And at this point, it's really critical for us to actually be able to measure the solubility of our compounds. And so we're adding now an additional screening or tier to our screening funnel. So through our collaboration with AstraZeneca, they provide in-kind ADME data, so absorption, distribution, metabolism, and elimination data. For every compound that we make across our projects, I cannot stress how critical their contribution has been to our research. So that really enabled us to actually focus on property optimization. How do we make our compounds more drug-like? And so NEU 1953 was our kind of front-running compound. We were moderately potent. We had improved our solubility. Both of these values were improved relative to our starting point, but we weren't really having a whole lot of luck in terms of improving the solubility overall. And so we really focused in on that aspect of the project. I won't go through all of these in great detail, but there are a number of strategies that are well known to improve solubility. Firstly, we can think about adding ionizable groups. The idea being that once the compound enters the body with the ionizable group, it will be more soluble. Unfortunately for us, it was not active against the parasite, and so that was not a viable strategy moving forward. We can also introduce an orthosubstituent, and so I'm demonstrating here with the introduction of a methyl group. The idea being here that we're going to introduce a kink in terms of the structure and prevent it from stacking, which can be a real problem in terms of solubility. Unfortunately, what we found was we increased our toxicity against HepG2 cells. Our third strategy was actually thinking about inserting an amine. So again, this introduces an ionizable element, but it also introduces a degree of flexibility and rotation that we didn't have in our original compound. But what you can see here is we actually had some really variable results. For some compounds, we really improved our potency, but not our solubility. For others, we saw a decrease in our potency, but an increase in solubility. And it was just really not a viable strategy when it was so unclear what our options were moving forward. And so what we found was the most successful was actually increasing our sp3 or saturated carbon content. We were able to install bridged piperazines like with NEU4443 and also the homopiperazine in NEU4363. The real breakthrough actually came serendipitously when we made, accidentally, NEU4438. We saw a significant improvement in terms of the potency. We were now at almost millimolar solubility. And we've really improved all of the metrics that we had kind of set out to achieve. And so this is now our kind of final screening cascade, if you will. So we're still generating all of the data against the various parasites. We've also now added Leishmania donovani, and we've actually changed our collaborator in terms of the cruzi data. So we're now getting all of that generated through UCSD. We've also had Tier 2 ADME come on board, thanks to a collaboration with Charles River Labs. And this has really meant that what we can do now is focus our limited research funds in terms of being able to do some of these later stage experiments. And so we now have this compound, NEU4438, that we can take through into PK, into an infection model, and then on in terms of further safety profiling and kinase selectivity. And so that's exactly what we've done. What we have found here is that, so this is a little bit messy, but in terms of red, red is NEU4438, and green is actually one of our other front-running compounds. But what we're showing here is for red, those compounds, sorry, the mice, each symbol on this image actually represents a different mouse. And so the images, the symbols in black is for the vehicle. So when we look at on day 3, and I'm hoping you can see my cursor, on day 3, we actually have a 168-fold change in terms of the parasitemia for NEU4438. Unfortunately for us, by day 7, when we stopped dosing, we actually then saw a rebound in parasitemia. And so we're actually doing further research now to see if we can understand why that is, actually look to see if we can identify the target of this series of compounds, but also thinking about can we optimize further, what criteria do we need to change to be able to affect a cure in this disease. Before I move on, so obviously I mentioned to you that we're obtaining data against all of these different parasites. So NEU4438 is our front runner for human African trypanosomiasis, but it's also incredibly potent against Leishmania donovani. And so that's actually a collaboration that we're still pursuing with the University of Georgia and also the University of California, San Diego. NEU5827 on the right is actually, that was originally identified as our best compound for PFAL through RARE. We're now actually refocusing our efforts on malaria and working with the University of Cape Town to do that. But we also have actives for T. cruzi as well as schistosomiasis through separate collaborators at UC San Diego. And what I wanted to really emphasize with this slide is that thanks to the collaborations that we have, we are really able to generate a significant amount of data, which identifies additional starting points for us to be able to launch new drug discovery campaigns for other NTDs and infectious diseases more broadly. And so what I hope I've been able to share with you very quickly is that drug repurposing is a viable strategy to identify lead compounds for a range of NTDs. And that by collaborating widely, we are able to obtain drug-like compounds from historical industrial programs. We're really able to focus and prioritize our resources, our limited resources in terms of our research program. And we're able to rapidly advance some of these projects. And I think also it's important to mention that distributed drug discovery, where we're working with industrial partners, we work with public-private partnerships like the DNDi, it's really essential for driving our NTD research. And that really just leaves me to thank the members of the lab who have worked on this project over the years. We have Dr. Baljinder Singh and Dr. Naresh Gunaganti, who are currently doing research within this space, as well as a new graduate student, Caroline Millard, who is also picking up this torch and carrying it forward. Our collaborators at the University of Georgia have been with us from the very beginning on this adventure, and we're very fortunate to be well funded by the National Institutes of Health, as well as receiving in-kind contribution from a number of other companies like AstraZeneca and Charles River Labs that I mentioned today. And that is what I wanted to share with you today. So thank you very much for your attention. For those of you who may be able to collaborate with us, we are always looking for potential avenues to collaborate. We really do focus on kinase inhibitors historically. And so if you have inhibitors of CDK2, PDE4, PI3 kinase, EGFR, or GSK3 beta, I would love to talk to you about that. So thank you very much.
Thank you so much, Lori. That was great. I know Julia had two questions, so I think maybe we can take the first one, which I think will be quicker, and then Julia, we might save the last one to the end if that's okay. So Lori, I think you can see it in the chat, but it was just the question is just a quick explanation of flat SAR.
Yes. Excellent question. So flat SAR really is referring to when a compound, we can modulate and modify the structure around that region, and we're not really seeing any changes in terms of potency. So it's pretty amenable to change. That is how I would explain that one. I hope that answers your question, Julia.
Thank you, Laurie. Yes, Maya said we'll get back to the next question after the other presentation. So yeah, Maya, back to you.
Okay. So we're going to move on then to our next speaker. So Dr. Kratz, if you would like to go ahead and share your screen.
Yep. Thank you, Maya.
Thank you.
Let me see if I can share my screen properly. Okay, there you go. Can you see my slides proper now?
Yes, perfect.
To get also here. Well, thank you again for the invitation and to have the opportunity to talk a little bit today about what the NDI has been doing in terms of drug discovery in endemic areas. So I'll talk a little bit about one or two of our projects in Latin America and how does that fit into thinking R&D for neglected tropical diseases in a distributed way, and also how can that impact the lives of the patients and also the scientific environment in several ways. I think my presentation is going to be much less technical than Laura's, and I hope that's all right. I'm going to talk a little bit more about how we do things and not really much about our projects, but I'm happy also to discuss that later on during the question. So for those who doesn't know, DNDi stands for Drugs for Neglected Disease Initiative, and basically the organization is a non-for-profit R&D organization, and can also be called like a product development partnership, a PDP, that was created back in 2003, and basically to fill in the gaps in terms of innovation in the pharmaceutical sector for neglected tropical disease. It was very clear that these diseases, they inflict a major health and social impact across the globe, and the unbalanced proportion of R&D investments and efforts in these diseases was very clear. So the NDI was creating, and other organizations similar to this were created back in those days in order to try to implement and create and implement a different model of R&D in order to fill these gaps, and they were not really fitting into the traditional pharmaceutical industry business model. So basically the NDI, in a nutshell, is an organization that raises funds from public or private donors, and work through partnerships across the globe. Partners from different sectors, academia, government, research institutes, pharmaceutical industry, and working all together with the common goal of developing new, safe, and affordable drugs for neglected patients. So basically, the whole model is based on partnerships, transparency, and patients' needs. So it's a little bit different from the traditional model in the pharmaceutical industry, but it does follow the same, more or less, rationale for developing new drugs. And it's important to mention that the idea here is really to bring the best science to the most neglected, and trying to do very high level world-class science for a different purpose here. So basically, the organization has this goal and way of working. I won't go into too much detail, but the NTDi has more or less 200 partnerships around the globe with these different actors I mentioned. And it has, since 2003 so far, developed and implemented 8 different new treatments for a couple of neglected tropical diseases. Some of them are listed here below. A lot of progress has been done in terms of new treatments for sleeping sickness, leishmaniasis, and Chagas disease, for instance. Also in the early days, malaria. And more recently, HIV, thinking about pediatric HIV development, and also hepatitis C, where not really the R&D is the issue, but more around pricing. So the access is limited due to the high prices. So the NDI is aiming to develop new affordable treatments in this field. And I think it's important to mention that to achieve these goals, the organization applied different approaches, such as repurposing efforts, such as Laurie was talking about, also development of fixed dose combinations, and also making drugs that are already available, although flawed and with sometimes severe toxic effects. They still have they are useful sometimes, and they making those available where they are not. But also trying to do let's say the end-to-end strategy of developing new chemical entities from scratch. And our aim is to, within the 25 years of our foundation, to deliver 25 new treatments. And for that, of course, we need a lot of partners, a lot of resources, and a lot of projects. And at the current moment, we have different projects and different development stages. And many clinical trials ongoing for these diseases, and as you can imagine, conducting clinical trials in the low and middle income countries in the middle of Africa or Southeast Asia or Latin America, it's not an easy task. So NDI puts quite a lot of effort in terms of developing these platforms and interacting with the local communities, and also trying to understand basic aspects of these diseases, and also trying to understand together with the patients and the scientific community and the clinical community, what does a new and innovative treatment for this disease actually represent. So really trying to build together what a target product profile is, and defining these goals for not only for our organization, but also for the community. So as I mentioned, we have a couple of different projects here, and the projects that I'm going to talk about, they are of course focused on the discovery parts of our portfolio, these long-term projects developing new chemical entities. And for that, the NDI usually applies a very similar approach that also Laurie was mentioning, we call phenotypic-based drug discovery. So usually we access large libraries of compounds through partnerships with pharmaceutical industries, or actually acquiring commercial libraries of small molecules, basically, and screening those compounds against the parasites or the different forms of these parasites in vitro, trying to identify active compounds, the so-called hits, and then seeding these hits after some initial validation and assessment of novelty and potential, and then seeding these hits into 3 or 4 nowadays lead optimization consortia that will bring different partners across different areas in the globe, trying to then optimize these hits according to several characteristics. Also comparing these properties in terms of potency, selectivity, physical chemical profiling, PK, and tox, in terms of trying to give these molecules the characteristics they need to go through the pipeline with a better chance of success. As we know, the attritional rates for infectious disease are very high and difficult to progress some of these projects. But this is more or less what we do. And we have these long-term, short-term, or medium-term new drug discovery projects based on basically phenotypic strategy. But also it's important to mention how the NDI see the way it works and how it can impact. Of course, the core mission of the organization is to develop new drugs, but it's also meant to showcase, let's say, the model in terms of like bringing the partnerships as the cornerstone of our mode of action. So we really interact with partners from different sectors and also different regions, including regions, endemic areas. So really trying to strengthen the capacities of R&D in these endemic areas. As you know, a lot of the areas impacted by these diseases do not traditionally develop their own drugs. Their pharmaceutical industries or even the whole science and R&D environment is not strong enough, or lack resources or particular expertise. For example, when thinking about Latin America, for instance, the pharmaceutical industry here is usually focusing on the development of generics, so it was not involved into a new radical innovation, as we call it here, developing new chemical entities. So it's important to bring these capacities here, but also to harness the resources of the region and leveraging the expertise of the local researchers. There's quite a lot of important expertise in the endemic areas from the clinical perspective, of course, being close to the patients, but also understanding basic biology, for instance. And also the NDI advocates for transparency in terms of costs, risks of R&D, but also open science and doing very, very much transparent projects and open innovation projects, something that I'm sure my colleagues will also talk later on about these aspects. So one thing I think it's important to mention, as I said, is that the NDI puts quite a lot of effort in developing these platforms in endemic areas. Initially, there was quite a lot of effort in developing these clinical research platforms. So we have the Chagas platform and the NENTDs, which are those focused on developing clinical development of Chagas and leash treatments in Latin America mostly, but also other networks in Africa focus on the development also leash, but also sleeping sickness and filarial nail treatments. This was very much part of the key initial steps of the NDI as an organization, really building the capacity needed to develop these drugs. But later on, the organization matured and also started looking into conducting drug discovery, this whole end-to-end strategy and bringing projects from all the stages of the portfolio to endemic areas with the goal of capacity strengthening and also leveraging the expertise and resources, as I mentioned. And one of the early, I think the first project that the NDI implemented in that sense was the lead optimization Latin America Consortium, a project that I've been involved for a couple, I think almost 7 years now. And this was a project that was really started quite small in Brazil with back in 2013. And this was very much one professor that had connections and someone knew someone in a conference, something really casual and informal like that. And then the NDI started this seed project funding a little bit the activities of the Professor Luis Carlos Dias at the University of Campinas in São Paulo, Brazil. With funding one chemist to start working on one of the chemical series that were identified within the NDI screening efforts. And this whole seed project had more or less two goals, as I mentioned, the technical goal, so really developing a drug discovery platform, identifying and optimizing these new chemical entities for Chagas and leishmaniasis, but also the capacity strengthening goal in order to organize really an independent and autonomous core unit, drug discovery core unit in the region that will be able to connect with other DNDi projects around the globe and in a two-way fashion. It's important to mention that this project started very small, but over the years it got support from multiple funding agencies and stakeholders in the region. Brazilian Health Ministry, also the National Development Bank supported our project for quite a while, and more recently FAPESP, the funding agency from the São Paulo state, is supporting our consortium in a 5-year grant. Over the years, this project, as I said, started very small, but it was able to map and incorporate new members and new capabilities. As you know, drug discovery is a multidisciplinary effort. You need chemists, biologists, parasitologists, people working with pharmacokinetics, tox, animal models, in vitro models, pure chemistry. So there's quite a lot of capabilities that are needed to progress these hits into promising leads, and then later on as preclinical and clinical candidates. So there was quite a lot of effort in terms of putting that. A lot of the capabilities were not present in the region, so the project was supported by international partners of the NDI very much in the early days, but throughout the years we were able to implement and validate these assays in the region, it was quite unique, and now we do have a platform like that. I'm going to show you more into details. And I think one thing that was key from the early days is that it shouldn't be like only a one way, so really bringing just capacity to Brazil, but it should be a two-way. So there was quite a lot of efforts in terms of putting these, connecting the project that was being implemented here with the global efforts from the NDI. This is very much a success nowadays. The project provides very important input to the NDI's global efforts. Within this structure nowadays, we were able to progress quite a lot of hit series into hit lead programs, going from early hits, actually not so much selective or potent in vitro, to partially active compounds in vivo. Several publications, all open access, were published, and also data sets that were made fully available to the community on these compounds. And more than 20 postdoctoral PhD researchers went through this consortium, and it's now quite a reference for the academic groups and also the pharmaceutical industry here. The NDI interacts quite a lot with the research community, trying to understand what projects are being done in the region and sharing resources and knowledge. So this is the structure we have nowadays. So the NDI works very closely with the team at UNICAP in terms of design and synthesis and coordination of the project. And then as soon as we get the compounds, we design and synthesize these compounds and ship them to different labs for different types of assays. We work closely with the University of São Paulo, where we run our first year parasitology and in vitro ADME assays. And we also have partners that are able to provide secondary parasitology assays, looking into mechanistic assays, also different strains, different panels of DTUs for Chagas, and also looking into more prioritization towards particular targets that are known or unwanted, for example. This is also in place here in Brazil, and also to conduct a lot of the in vivo pharmacokinetics and toxicology, early stage toxicology studies, also through partnerships in the region. And then later on, when these compounds progress, they screening cascade and reach certain cutoffs in terms of target candidate profiling, they would move on into the in vivo animal models of these diseases, which is still being done outside the region, but we have efforts in the moment to implement and validate these assays to bring more or less full circle these initial drug discovery lead optimization efforts. Again, we still rely quite a lot on the support from international partners. Nobody does drug discovery alone, but it was part of our objectives to bring as much as possible to the region. This is the screening cascade we use for our project. I won't go into much details, but it's important to mention, as I said, this is a phenotypic-based drug discovery campaign. So we do rely quite a lot of screening these small molecules against parasites, intracellular forms of these parasites, but in parallel generating as much as information we can in terms of physical-chemical properties and also ion properties of these compounds, trying to reach as fast as possible to the exposure and then progressing these compounds for in vivo proof of concept studies. For those who are interested in this project, this is the LOLA project is part of a larger consortium supported, as I mentioned, by FAPESP and in partnership with Medicines for Malaria Venture. We have this website, there we have job opportunities for people willing to participate in this consortium, for example, and also we share knowledge and news about the drug discovery projects in the region. And in fact, DNDi has established what we like to call like this virtual orchestra of partners in the region, covering different expertises and different partners as from academia, but also more like CRO type of small startups and organization, and also pharmaceutical industries in the region. We recently started a new project of what we called SPDD Leash, which is a structure-based drug design. So we are starting to expand our scope of strategy, also looking into more, as soon as the new targets become available from some of these parasitic diseases, especially Leishmania. There was quite a lot published recently in this region, in this field. We're starting to look into developing target probes that will help us validate chemically and genetically these targets in these parasites, and might drive some of the drug discovery projects later on downstream on the pipeline. And also, as the project matures and there's more resources, more people, and more knowledge concentrated in the region, we're starting to explore different collaborations with other partners in the region, and small projects are starting to bloom, which is quite interesting, and very, very proud of what we're able to achieve in the past seven years in the region, trying to contribute to the best science, to providing valuable input to the NDI global efforts in terms of drug discovery, but also to interact with the research community, strengthen the capacities here, and build this nice environment for science around neglected tropical diseases in an endemic area, which is quite important. So I'd like to thank you, and I'll be happy to answer any questions.
Thank you so much, Dr. Kratz. So I think we'll hold off on the questions until the very end. And so we can get to our final speaker. So that was really wonderful. And I particularly loved hearing about the collaborations and how collaborative. I mean, I've been a fan of DNDi's for a long time, but I love the focus on collaboration. I think that's so important, especially in our field. So I'm excited to learn more through the Q&A session. But at this point, I will turn it over to Dr. Klug to finish off our speaker panel.
Well, thank you, Maya, and thanks for the invitation to talk here today. Let's see. Just going to share my screen. Right, so my name's Dana. I'm a postdoc in the Todd group at University College London, and I'll be sharing a little bit today about our open approaches to developing new medicines. So what really we're trying to do with all of our open projects is to take drug discovery, which can be this highly multidisciplinary, and I think, as Lori and Jadel both have highlighted, highly collaborative process. So sort of taking the whole idea of drug discovery and making it open source. And so what I mean by that, I think, is sort of encapsulated pretty well by the 6 laws of open source drug discovery that Matt published in 2019, which sort of set out some of the principles that all data and ideas are freely shared. Anyone can participate at any level. We don't pursue patents. We encourage suggestions and public discussion, and an open project is bigger than and not owned by any given lab. So the idea is to make all data and ideas as accessible and as open as possible. And so there are a lot of advantages to doing drug discovery this way. So we can quickly find effective solutions to problems by recruiting participants with a diversity of expertise. So we've had experts that are working at pharma companies chip in. We've had participation by high school students. We can avoid duplication of effort by openly sharing our approaches, including failed ones, so we can learn just as much from approaches that didn't work as ones that did. And by sort of sharing them openly, we can direct our resources to more productive approaches. And it's an opportunity to address problems like NTD drug discovery that are not well served by a patent model, because everything's in the public domain in real time. we're sort of precluded from pursuing patents for these things. And so it makes it much easier to share solutions when we develop new chemical entities. So how does this work in practice? We use a lot of online infrastructure to make open source drug discovery work. We use electronic lab notebooks that are updated in real time. So if you ever want to see what experiments are running in a project, you can open up the ELN. Those are shared publicly. We have open project meetings. Anyone who's interested can attend these. We record them and we put them on YouTube. So they're accessible if you want to watch them back later. We use the GitHub platform for discussion and for keeping track of the status of the projects. So all of the discussion is in the public domain and it's online. And the funding, we're typically funded like a lot of other academic drug discovery groups through grants. We also get some in-kind contributions. And everything defaults to this Creative Commons license, which is the goal is to make sharing information as simple as possible. So as you can imagine, this approach works very well for NTDs when you have a situation where you need to quickly develop cheap and effective therapies for these diseases that don't attract investment from for-profit pharma companies that tend to be patent driven. And so it's a situation where it really benefits from maximal sharing of resources and information. And we do work with organizations, as Jadel talks all about, DNDi and MMB as well as our academic and other collaborators. So I just thought I'd give you some highlights from some of our open source projects. So I would say open source malaria is one of our bigger projects. We've had several publications come out of this work, including this one in ACS Central Science in 2016 that describes some medicinal chemistry that was done. We have had participation from artificial intelligence companies in predicting activity of drug candidates. And we've had some very nice work on the biology side as well, exploring new targets for antimalarial drug discovery. We also have a program looking at mycetoma. So this is a fungal infection. We ran a high throughput screen of the pathogen box and the stasis box from, these are from the MMB compound boxes. And we are pursuing 3 different chemical series for mycetoma. And this is another project that nicely highlights the community involvement. So we've had some synthesis being done by a high school here in the UK. And they're synthesizing some new compounds that we can test for mycetoma activity. It's not technically an NTD, but I thought I would tell you a little bit about our antibiotics as well. Similar to NTDs, the economic incentive to develop new antibiotics is low. And so it's another opportunity for open source drug discovery because there really is a need for new antibiotics due to the rise of antimicrobial resistance. Like MMV and DNDi, there are organizations and platforms around antimicrobial drug discovery in the community aimed towards sharing research, sharing resources and knowledge such as COAD, SPARC, and GARD-P, as well as us, we are OSA. And so some of the highlights from this are our fragment screening, which we screened fragments at Diamond XChem against these MER ligase enzymes. So our goal is to develop a compound that inhibits multiple MER ligases across multiple bacteria, so we can develop a really broad spectrum antibiotic. For the MER ligase project, we've also had a contribution from a company called Atomwise that uses machine learning to design de novo inhibitors. So these compounds have been synthesized and we'll test them against the MER ligases as well. We also have another series that's a phenotypic assay where we're seeing some activity against MRSA and trying to optimize the clearance and solubility of these compounds. So I think I went through that pretty quickly, but I tried to just sort of hit the highlights of our open source drug discovery, and I'm happy to take questions on details. I'd like to acknowledge the rest of the TOG group, and for all of our projects, I've tried to acknowledge people whose work I presented on the slides, but of course, because it's an open project, there are many other contributors to all of these that make a huge difference. So if you'd like to learn more or get involved in any of these projects, the GitHub page, I think, is a good place to start. So each of our projects have their own page. We also have Twitter feeds for all of these if you're interested in more periodic updates. And of course, you're welcome to get in touch with me on Twitter or via email as well, or I'll answer some questions now. So thank you very much.
Okay, wonderful. Thank you, Dr. Klug. Again, I think the fact that it's all open source and, how all three of you have shown that, you're looking for collaborations, you're open to collaborations. I think that is so important because, what we've been talking about today has been in part how maybe it's not so far, maybe we don't have so far to go to find really alternative treatments for some of these diseases. And it's really just coming together and reworking some of the things that have already existed, or just taking up things that have been, abandoned because they weren't profitable, marketable, or, for the right population, right? So I think at this point, I will we'll open it up to any questions. I think Richard has been monitoring the chat for those. So I will turn it over to Richard to run the Q&A session.
Yeah, thank you very much, Maya, and of course, all our speakers. We are grateful to you for sharing your time with us. There are 3 questions to start with, and a couple of questions to Dr. Ferrins, and then I'll start with this one just to end on the point, Maya, you were remarking regarding the importance of collaboration. This one is from Joseph to Jadel, asking, even investigator has a need for preclinical ADME assays for a lead compound. How can they receive support from LOLA? Is there an application? I think you were speaking to that at some point, Jadel. Maybe that's a quick clarification question.
Yeah, sure. Yeah, I think I'm afraid I don't have like a very straight answer. I think there are several options here. We do support informally, let's say, other investigators working, let's say, a particular series or a particular target, for example. Sometimes we do support these let's say, demands in a very informal way, without any collaboration agreement or transfer of resources, anything like that, just providing access to the assays and the data as fast as possible. But if things are getting more, let's say, structured and the need is higher, and it's also there's more potential, there are several ways that we could do. We could just point towards the groups that are part of our consortium and see if the other group, for example, they want to collaborate in separate outside of our consortium, but we can also consider working together and internalizing this series, for example, or a project after thorough analysis and discussion with the different partners. So there's different ways to do that. And so the platform that we developed, I mean, it's part of a DNDi project, but it's also groups that are conducting their own independent projects and that they can interact with the community freely.
Thank you, Jadel. I'll go to a question from Julia. There are 3 questions she's posted, and this one specifically is to Dr. Ferrins. So, and I hope I can capture the message with a question. She was asking about a compound that lets parasitemia rebound after dosing is stopped. Is low toxicity, so could you combine it with another one? For instance, using that second one also at lower dose, like fexindazole. She also asked, thereby decreasing toxicity for both while maximizing antiparasitic activity. And I think also followed up with the response you are making about flat SAR, whether that's a good thing or not. I hope the question comes through, Dr. Ferrins.
Yes, thank you, Richard. So to tackle the first part of that question, so co-dosing, yes, it's certainly something that we could consider. It's definitely precedented. You've got nifurtimoxiflornithine combination therapy currently used to treat HAT. And so that's definitely something that we can pursue. And our efforts with that compound have not stopped. We're still looking to see if we can optimize dosing regimen to actually elicit a cure. There's a number of different things that we can still attempt to work with in terms of improving the potential outcome for that compound that I think you're referring to. So yes, co-dosing is definitely an option. And then I'm sorry, I missed the other part of the question. Was a follow-up on flat SAL? I'm just scrolling through the chat to see if I can see it.
That's correct. Yeah, Laurie, was whether it's a good thing or a bad thing.
Oh, a good thing or a bad thing that you have flat SAL? I mean, I would say it's a good thing in a way, because it means that you can modulate that area of a compound, and you can use it as a strategy to try and improve solubility. And I didn't show that to you today, but it's certainly something that we did do. We tried to really take advantage of that area of the compound by thinking about how, what could we put there that might impact the properties rather than being potency driven, because we weren't seeing big differences in terms of our potency profile in that region. So I guess you could say it's a good thing. It could be considered a bad thing if I flip that around, because it means that whatever the target is, it's likely that you're not getting meaningful interactions between the compound and the target of interest. Now we don't know what the target is of that compound. We are looking to see if we can understand it, and then actually use structure-guided drug discovery to try and optimize further, or to see if we can understand what's happening in terms of the binding mode of these compounds. But that is a long and lengthy process that we have just begun. So I hope that answers your questions.
Seems she's pleased with that. Yeah,
I think so.
Yeah, there was two other questions, and I think well, let's get to that. So Julia was also curious about whether, and this was to you, Laurie, about explaining a little bit about open science and open discovery. She says because it must be so different from the current academic industry paradigm. Is that so? Please, Laurie.
Sorry, was this to me or to Dana Klug?
It was to you, but I think it can be. Both of you can respond to that question. Yes.
Okay. Could you just repeat the question for me again, please? Sorry.
So right, yeah. Explain a little bit about open science and open discovery, because she supposes it's going to be different from the current academic industry paradigm.
Yeah, I think it's definitely a different mindset. However, there's definitely advantages. It's certainly something that we as a group have we've been thinking about and actually learning from the Todd Lab and Dana's research efforts in terms of actually trying to improve our openness, if you will. While we are very happy to collaborate with whoever, we certainly don't ask for material transfer agreements or non-disclosure agreements. We're happy to collaborate openly in that fashion. But we haven't yet made that jump to being able to do it, and so that it's in the public domain and everyone can kind of contribute. It's certainly something that is a different mindset, but I think that Dana would probably have something more to say on this.
Yeah, I could chime in. I think, actually having worked in Mike's lab at Northeastern, where it's definitely a lot of industry academia partnership, I would say that actually open source is not that different. The only difference is that obviously everything's in the open, but obviously we have had some buy-in from industry for, I would say, probably the same motivation as they would collaborate with someone like Laurie, which is that they do get some good PR out of it, and also they sort of raise their company profile by participating in NTD drug discovery or an open project. So it does have benefits for pharma companies as well. And so, sometimes people can be hesitant, but usually we're able to address whatever questions they have about participating in an open project. And so I think in my experience, it works very well. And we're able to have the same kind of discussions and things that, you would maybe have even if your project's not open, we are able to have those in the open for the most part. Without any sort of hesitation, once people get used to the idea of just this is happening online and people can see it.
I think, sorry if I can just jump in. I think actually if Jadel could also comment on that, because I think you talked a lot about this.
I can also provide maybe different angles to that discussion. I think it's very important for those willing to do it and hasn't done yet, for example, an open project. As soon as you start trying, you'll see that how much faster and cheaper and more diverse the project can be. The partners, they provide different resources. Sometimes these are in-kind resources, and also provide different expertise into the project, and that might accelerate and improve the quality of the research. I'm pretty sure that's the case for a lot of the more open exploratory projects that at least the NDI has worked with. Some are already mentioned here. For example, the results we obtain through sharing compound tools, but also providing negative data, as Dan already mentioned. This is quite important in order for others to learn and also use the data thoroughly in modeling, for example. You see, for example, the success of projects such as the COVID moonshot, for example, recently working with these protease inhibitors in an open source way. It's amazing how fast they could progress these compounds and go close to a clinical candidate working fully in the open. I think from the moment the industry and the academic labs, they start learning from the examples that are out there and actually doing it, they will clearly realize the power of conducting open science.
Right, thank you very much, Jadel. There's a couple of questions. One is from Matthew Todd, and the other one is from Crystal Lee. Let's go with the Matthew Todd question. He's saying that I'll be interested in knowing the thoughts of people here about whether there are regional differences in how people view open science. Are there places that people think are more or less receptive to open science, or whether everywhere is pretty much the same? Seems that's a good question in context of what the conversation has been so far. I wonder if you've tackled that, or that's a conversation, open conversation, Jadel, Laurie, or Dana, about are there differences in this question?
Very good question. I'm not really sure I have the answer. Maybe different sectors have different views. I mean, sometimes I would say pharmaceutical industries, let's say, I could talk about a few examples or regional experiences. So while some companies were like starting the process of drug discovery, starting to explore this more radical innovation, and they decided to keep it close and trying to protect others, were viewing it as the open science as an opportunity to learn and expedite the process. So it's very hard to say that, let's say, one region will be different than the other. I would say I would still think it will be case by case, and also how much the stakeholders had the chance to participate in other projects or seeing good cases to be more pro or con open innovation. I'm not sure if this can make any sense at all.
So I would tackle that question in a slightly different way, I think. And I guess for me, I'm thinking about it from my PhD, I did that back in Australia. I have worked in the United States now for the last 6 years. And so my experiences are very, focused in those areas. I wonder if there's an element of, and I don't know how to say this in a in an appropriate way. So I'm just going to go for it, which is kind of my nature. I feel like there's a level of understanding perhaps in some regions of the world that not all of the information, not all of the skills that you need are in that one institution. And I think that that is something that maybe other countries, I don't think the United States is necessarily like a great example for this. I feel like, you know, in the United States, we're very insular and we have a tendency to be kind of, you know, we can access what we need through our network and not kind of expanding out. I kind of feel like LOLA in a way is a really great example of that, of this kind of idea where it was, you know, DNDi were bringing together the different elements of people to kind of facilitate a drug discovery program. And I wonder if it's a similar kind of mindset, acknowledging that, you know, one institution cannot drive a complete drug discovery program. And maybe it's also got something to do with the disease area that you're working in. I think NTDs is really very amenable to open drug discovery and under-resourced diseases in general. I know Dana mentioned antibiotics. There's also, you could imagine, for antifungal agents, a similar kind of mindset, because whatever you make is not going to bring in a significant revenue that would kind of, I think, interest a pharma company. And so I think to Jadel's point, it's completely right. I think industry is going to be very concerned and driven by, you know, what is patentable, because that's where they make their money and are able to kind of please their shareholders and do all of those sorts of things, which they need to in order to be able to kind of survive and to compete. And that's, I think, how I will leave it there.
Yeah, well, all of my experience with open science has been while working with Matt in his lab. Hi, Matt. So I think I would agree with what Lori and Jadel said, and would also maybe add that in terms of academic scientists in open drug discovery, you can sort of see a generational, I think, push for open science, where a lot of younger PIs or early career investigators really are on board with the idea of doing open science, and that including in the drug discovery field. So I think that's very cool as well.
Right, thank you very much. There was one, I think it was a clarification question, Dana, to you from Crystal. She was curious if you could elaborate a little bit more about how the machine learning you use works. I think you mentioned that, does it use 3D modeling, or is it more along lines of analyzing assay permeability or bioavailability, etc.?
Yeah, so I am not by any means an AI or modeling expert, but what I can tell you is that, so the way that this was done was that we actually ran a little bit of a competition. And so in general, what we said was, here's the data that we have for all of these antimalarial compounds, the structures, their potency, their solubility, basically, because everything's open, they have access to all of it. So you can feed this into whatever model that you want to build. And what we want to know is, by doing that, can you suggest structures for us to make, and will they be potent antimalarials? And as a secondary consideration, it would be really great if you could help us improve the solubility and the ADME properties as well. So we had several companies participate in that, as well as some academic groups. Some of them used, I believe some of them used target-based modeling. Some of them didn't, and I've linked the preprint of the write-up to that, and of course everything related to that is on the GitHub page as well. And so they submitted their compound designs. We synthesized these compounds and then tested them. And so some of them actually did turn out to be potent inhibitors, which was very cool. But I would recommend the paper if you want details about the actual models themselves. So I hope that helps.
Thank you very
Okay, thank you very much. So we're right at time. So I think there are no more questions. So I want to once again thank our panel so much for joining us. And I think this was really great. We will be sending out the link. It will be posted on our YouTube page as well, along with last month's webinar and any events going forward. So we will be having one at the end of April as well. We'll be looking at surveillance in NTDs. So we really encourage you to join that. I'm also putting the link to our newsletter again in the chat in case anyone missed it. So please do sign up if you'd like to be aware of any of our other events and things going on. We also highlight a disease every month and put some relevant research about that and highlight other grant opportunities and as much as possible. We call ourselves the New England NTD Consortium, but really we are global, like all NTD work I think is and should be. And so we welcome anyone. So please don't be put off by the name. Our initial ambitions were small, but we do hope to grow globally. So again, thank you everyone so much. And we look forward to seeing you at our next event. Thank you. Bye-bye.
Thank you.